Okay, before we dive in, just a quick but really important note. This presentation is a summary to help you understand the ICH M14 guidance. It's absolutely not a substitute for reading the full official document yourself.
Welcome to Evidence Mastery. I'm Dr. Masoud Tusi. If you work with real-world data, you understand just how crucial it is to make sure your studies are solid, robust, and accepted globally. And that's exactly why today, we're going to break down a true landmark document that sets the new standard, the ICH M14 Guidance. Now that's a mouthful, I know, but the full title tells you everything. This is the International Council for Harmonization laying out a single, high-level standard for how we should all be using real-world data to assess medicine safety. The goal here is to harmonize the approach across the entire globe. So, we're going to walk through this in a really logical way that actually mirrors the life of a study itself. We'll start with the scope and purpose, then look at the core framework, design, protocol development, analysis, and finally wrap up with dissemination. First up, Section 1, Introduction and Scope. This is where the guidance really sets the stage and lays out its fundamental purpose and vision.
The one big idea driving M14 is harmonization. The goal is to get everyone on the same page, with one globally recognized set of principles. So why is that so important? Well, it means a single, well-designed study can, in theory, be accepted by regulators all over the world. And that's a total game changer for efficiency. It streamlines everything, helps us avoid redundant work, and ultimately, it strengthens the evidence we use for critical regulatory decisions.
Okay, let's move on to Section 2, which gives us the conceptual framework. I want you to think of this as the master blueprint for the entire study process, from start to finish. As you can see, the guidance lays out a very clear stepwise life cycle. It all kicks off with a super precise research question. That question then drives a critical feasibility check, which is followed by protocol development. And this all happens before the study even begins. But here's a key detail. While this looks like a straight line, the guidance makes a point to say this is an iterative process. You're constantly refining and circling back at each stage.
And that brings us perfectly to Section 3, Initial Design and Feasibility. This is that foundational stage. Before you even think about writing a full protocol, you have to do your due diligence and rigorously assess if this study is even possible.
And this slide asks the million-dollar question of that feasibility stage. The guidance introduces this really critical concept of fit-for-use data. It's a simple idea, but it's profound. It's not enough that the data just exists. It has to be relevant and reliable enough to actually answer your specific research question.
So a proper feasibility assessment is like a pre-flight checklist. You systematically go through these questions. Can we actually find our population, our exposure, and our outcomes in the data? Are there even enough patients to get a statistically meaningful result? And, maybe most importantly, do we have the data we need to control for key covariates? Answering these questions early on saves you from wasting a ton of time and resources on a study that was never going to work.
All right, so once you've established that the study is feasible, we get to move into Section 4, Protocol Development. This is where the abstract plan becomes a concrete, highly detailed document. Now, the guidance makes a really important distinction here, starting with the conceptual definition. This is the what. It's what you want to measure based on clinical reality. For instance, think of myocardial infarction as a cardiologist would understand it. And then you have the operational definition. This is the how. It's how you're going to actually measure that concept using the specific codes and fields you have in your database. It's the practical algorithm that tries to translate that clinical concept into something a computer can find. And let me tell you, the gap between these two definitions, that's where a lot of errors can creep in.
And this table really illustrates the trade-offs you have to make when choosing a data source. On the one hand, you've got EHR data, which is packed with rich clinical detail but can be messy and fragmented. On the other, you have claims data, which is fantastic for tracking patients over time, but often lacks that deep clinical information. Understanding these pros and cons is absolutely essential for picking the right data to bring your operational definitions to life.
A great protocol doesn't just describe what you're going to do. It anticipates all the things that could go wrong. The M14 guidance really stresses that you need to pre-specify exactly how you're going to tackle these major sources of bias and confounding. It means you're proactively building the solutions right into your study design and analysis plan from the very beginning. So with that robust protocol locked in, we can move to Section 5, which is all about the execution phase, from managing the data and running the analysis, all the way to writing that final report. I love this workflow because it shows just how structured the process is. But the real hero here is the pre-specified statistical analysis plan, or the SAP. This is what keeps the science honest. It prevents things like p-hacking or fishing for results, and ensures that your analysis is rigorous and was planned out well in advance.
And this brings us to another powerful tool that the guidance emphasizes: quantitative bias analysis. This isn't just about saying, well, bias might exist. It's about actually modeling what its impact could be. How different would my results look if there was some unmeasured confounding? It gives you critical context for interpreting your final results.
Finally, we've made it to Section 6, Dissemination and Documentation. The study is done, the analysis is complete, but believe me, the work is not over yet. For real-world evidence to be truly trustworthy, it has to be transparent and reproducible. And that means registering your protocols publicly before the study starts, following established reporting standards, and keeping meticulous documentation. The whole point is to create a crystal-clear audit trail so that anyone can understand exactly what you did and why you did it.
And that brings our high-level walkthrough to a close. We really hope that you found this summary of the key principles in the ICH M14 Guidance to be helpful for your own work.
And just to circle back to where we started, remember, this was an overview. To really get this right and implement these principles effectively, we strongly encourage you to go read the full guidance document in detail.
And now, I'd love to hear from you. What are your biggest questions? What are the challenges you're facing when you try to implement the M14 guidance in your own studies? Please, drop your questions in the comments section below. I'll be reading through them and answering them myself.
If you found this explanation valuable, please subscribe to Evidence Mastery. We're going to be doing many more deep dives into the critical guidelines and methodologies that are shaping our entire field. Thanks so much for watching.